Hi, as you remember in the last video I have uploaded about the basics and fundamental concept of biomarker. And today I have selected the specific and hot topic in biomarker, that is genome biomarkers. So in terms of definition, a genomic marker, which is also called as a genetic marker, is a specific identifiable DNA sequence or genetic variation at a known location in the genome that can be used to track inheritance, identifying individuals or population, and associated genetic variations with biological traits or diseases. In simple terms, a genomic marker is a signpost in the DNA that helps scientists to understand how genes vary, how traits are inherited, and how diseases may develop. So, this is the fundamental definition and background of genomic biomarkers, where to identify the interpretable gene marker associations with functionally informed kernel-based tests in 190,000 exomes have been shown here. Now, you can see here three major things: gene annotation, observed variants, then qualifying variants, effect prediction, functionally informed association tests. And out of these things, this particular approach gives 30 biomarkers. But depending on your disease and disease specific conditions, you can develop more or less number of biomarkers, as you can try for the publicly available data sets. Now it comes to the key characteristics of genomic biomarkers. So, as you understand already, it is measurable. DNA RNA features like mutations, gene expressions, or some other things, for example, copy number alterations, structural variants, and that could help in diagnosis, prognosis, or treatment guidance. Of course, there is a need for validation for sensitivity, specificity, and reliability to enable the personalized therapeutic approach by linking the genetic profiles to the outcomes. The key features include reflecting biological processes like normal versus pathogenic or diseased, being quantifiable, and offering insights beyond the traditional biomarkers for the tailored therapies. So it must be reliable, safe and easy to measure, sensitive and specific, can be applied to diagnosis, staging, prognosis, and it should be the cost efficient for follow-up treatment or diagnosis and modifiable with the treatment. So a good genomic biomarker has the following properties like polymorphic, which shows variation between individuals, stable, inherited consistently across generations, detectable, can be identified using molecular techniques, genome specific, located at a defined position on a chromosome, functionally or statistically associated with traits or diseases. Now, after core characteristics, after this key characteristics, you need to think about the core features or characteristics. And here it is molecular basis, clinical relevance, predictive power, early detection, quantify. Molecular basis derived from DNA, such as SNPs, copy number variations, mutations, or RNA expression levels, and splicing, providing deep molecular insights. Clinical relevance indicate normal biology, disease presence, progression, or treatment effectiveness. For example, oncogenic mutations guiding cancer drugs, predictive power, identify the patients likely to respond to specific therapies or experience toxicity, enabling personalized treatment. Early detection provide data faster than clinical endpoints like mortality, allowing for earlier intervention. Quantifiable, objectively, measurable through analytical and clinical variation is crucial for the reliability. Now, the essential qualities for the uses of genomic biomarkers. It is like it includes sensitivity and specificity, that means accurately detect true positives and minimize the false positive rates. Reliability and reproducibility, consistent results across different tests in different labs. Practicality, feasible for clinical use, often requiring advanced tech like next-gen sequencing. Then clinical utility, must demonstrate improved patient outcomes, not just biological changes. And here you can see the basics about these genomic biomarkers, especially in cancer. So cancer biomarkers could be DNA, proteins, RNA metabolites, and could be found in tumor biopsy, urine, blood, saliva, stool, ascites. Then it could be analyzed by using FISH, sequencing technologies, immunoassays, microarrays, PCRs. Now, there are some well-known examples of genomic biomarkers. For example, in breast cancer, PTEN, STK11, ATM, PIRIP1. Then lung cancer, EGFR, ALK, FRET, MET, FGFR, DDR2, CTLA4. Colorectal cancer, KRAS, CEA, KIM1, Spondin 2, DCR3, TREL-R2, REG4, MIC1. But if you want to see in different subsections of genomic biomarkers, for example, in terms of gene mutation, then BRAF, BRAF, KRAS in cancer, guiding targeted therapy, gene expression level, in terms of gene expression level, miRNA, mRNAs, levels reflecting cellular states, epigenetic changes, for example, modifications to DNA impacting gene function, then germline variants, TPMT, or TYMS for drug metabolism. So now it comes to the types of genomic biomarkers. For example, SNPs or single nucleotide polymorphisms. A variation at a single nucleotide position, for example, A to G, G to C, or C to A, something like this. Most common genomic markers in the human genome you need to know. Then, what are the widely used GWAS, pharmacogenomics, and population genetics approaches for discovering these SNPs? Then, example, SNP in the CYP2C9 gene affecting drug metabolism. In general, in molecular biomarker section, it comes to the subsection in nucleic acid like DNA, cfDNA, ctDNA, RNA, mRNAs, miRNAs, LNC RNAs. Then in cells, you go for CTCs, proteins, antibodies, and autoantibodies, antigens, enzymes. Then it comes to the metabolites. So next comes to the short tandem repeat. That is STRs or microsatellites. That is another subpart of genomic biomarkers. So STRs or short tandem repeats, their sequences ranges from 2 to 6 base pairs repeated multiple times, highly polymorphic, used in forensic identification and population studies. So these STRs or short tandem repeats are also called as microsatellites, are sections of DNA with shorts 1 to 6 base pair sequences repeated consecutively like CAG, 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 something like this. Their high variability in the number of repeats between individuals makes them powerful marker for DNA profiling, crucial in forensics, paternity testing, genetic research, and identifying the disease links. And right side shows you the STR analysis examples. Now it comes to the copy number alterations and copy number variations. So these CNVs are the segments of DNA that vary in the number of copies between the individuals, involving duplications or deletions ranging from large chunks of 2000s of bases, while many CNVs are harmless and contribute to normal human diversities. Others can impact gene expression, leading to traits susceptibility to diseases or genetic disorders like syndromes, making them crucial in the evolution in medicine. These copy number variations can be seen here in the left side figure. This is the chromosome where you analyze the insertions, deletions for copy number. So what are the changes in terms of positive and negative way, like the addition of certain components to the reference genome, and with respect to the reference genome in case of when someone is developing some kind of diseases or when there is some kind of deletions. So this is how you can go for example, here genome, this is genome what you are saying, then target regimen, then SNPs, then copy number variations, which are generally quite bigger. So here you can see these are the copy numbers. And then sort reads, for example, one pair, then you can go for detailed mapping for all the chromosomes. This is, for example, one of the chromosomes, but you can check all the chromosomes in that particular organism. If it is human, then all the 23 chromosomes here you can see X and Y, and then 1 to 22 chromosomes. So this is what you can check it for any kind of the certain segments addition or deletions of the sequence, and then go for the final analysis in terms of variations. So this is the third types of genomic biomarker. Then it is comes to the fourth type, that is insertions and deletions, which is called indels. So normally it is a small DNA insertions or deletion and can alter gene function or regulation. Insertions and deletions are fundamentally genetic mutations involving the addition, that is insertion, or removal, deletion of one or more DNA nucleotides, leading to the gaps in the sequence alignment, significant sources of evolutionary change and crucial for genetic variation, disease, and evolution, often occurring as single base changes, but also larger structural variants impacting protein function, especially when they alter the read frame or frame set. So in general, you need to remember that it is a small DNA insertion or deletion and could alter gene function and or regulation. So this is the example showing for the indels or insertions and deletions. So here, this is DNA representation. Then a standard method for analysis is GATK, SAM tools, etc. And then you look for the insertions, and you can also look for the deletion part. So here, Pindel or de novo alignment, and then you go for mapping, and then you have to compare with the reference genome. Now, next, fifth type of genomic variation is the structural variants, which is normally the variation in the structure of an organism's chromosome structure. So you need to remember that there is variation in the structure of the chromosome, such as deletions, duplications, copy number variants, insertions, inversions, and translocations. So here you can see this is the reference chromosome in this top figure. So, and then after deletion, the chromosome is slightly reduced in size, but after duplication, it is increased. Then there is insertion. There is again change in the structure of that specific chromosome with respect to the reference unit. So, what it could include in structure, variants or variation, deletion, duplication, inversion, insertion, translocation. But what could happen when you so normally because of these things there must be change in the structure of the chromosome. Now, what are the method how to identify the genomic markers? So for this, you need to understand the computational approaches because this is the mandatory part when you are going to identify the genomic markers. So there are a number of technologies, machines, and computational approaches. Now, in terms of the technology, they are detected by using the modern molecular and computational techniques, which could include next generation sequencing or NGS, whole genome sequencing, whole exome sequencing or WES, then genome wide association studies or GWAS, GWAS. Then it comes to the computational pipelines, that is bioinformatics pipelines called variant calling, annotation, and many more steps are required. You can see the publications from my side also in my Google Scholar. You can find a number of research and review papers which will help you to understand how to identify the genomic markers. Now, you need to understand that genomic markers are identified by analyzing the DNA samples. From where? It could be from blood or swab or somewhere else, depending on your target. Using the molecular techniques such as PCR, sequencing, and gel electrophoresis, which detect variation in DNA length or sequence, revealing polymorphic sites used as landmarks to map the genes or track the inheritances, often involving the comparison of individuals' DNA to find the differences linked to the diseases. Now, there are the pipelines. There are a number of pipelines. For example, someone can say we can go for one of the application where we can predict the biomarkers and go for precision medicine or personalized therapeutics. Then, here in this pipeline, you can see how to collect the biopsies for predicting the biomarkers and then how to predict the compatible drugs for that specific biomarkers by using computational approach. After predicting the in silico step, the biomarkers, you could also go for the laboratory testing or validation. Depending on the quality of prediction computational approach, the validation could vary. So if your approach is quite hard and fast, then there is always most often maximum chance of experimental validation also. Now, you can think about purely computational based prediction where you can go for multimodal AI ML for discovering the novel biomarkers and predicting the diseases using multi-omics profiles of the patients with, for example, cardiovascular disease. This is from Nature Review paper. So you can look and read this paper and you can find the detailed application of each of the steps involved in this AML based AI ML based or artificial intelligence or machine learning based approach. Now, the next approach is synthetic biomarkers, which is also now becoming promising. And the title of this review paper is the same like Synthetic Biomarkers, a 21st Century Path to Early Cancer Detection. And this is again where you can look is Nature Reviews Cancer. This is the journal. And here you can see itself from this graphical abstract that how you can proceed for the biomarker discovery and early cancer detection steps. Now, the next step could be since there is so many promising points with biomarkers, then there is also a big impact in the market growth. So, biomarkers, market growth, drivers, and opportunities are huge. So the trends and disruptions influencing the customers, so customers. So here you can see how they could impact your financial dealings also or businesses with when you are involved with biomarkers. So this is also the market value of biomarkers. Now it comes to the biological and clinical significance. In terms of biological and clinical significance, it comes to the disease risk prediction. For example, genomic markers could help to identify the individuals at higher risk for different human diseases, cancer, diabetes, cardiovascular disease, neurological disorders. Or you can go for pharmacogenomics approach where you can predict the drug response, drug toxicity, and optimal doses for each therapeutics. Where pharmacogenomics is the science of how your genes influence your response to the drugs, combining pharmacology and genomics to tailor medication for better effectiveness and fewer side effects. Moving beyond the one size fits all approach to the personalized medicine. It helps the doctors to predict if a drug will work, find the right dose, and avoid adverse reactions by understanding your genetic makeup, which affects how your body absorbs and metabolizes and uses the medications. This leads to a safer, more efficient treatment approach for conditions like cancer, heart disease, and Alzheimer's. Next, it comes to the application where you can go for is population genetics and evolution. You can think for genetic diversity study, trace the ancestries, and understand the evolutionary relationship. So, population genetics studies genetic variation within and between the population. It includes so population genetics normally you can look for the variation within or between the populations, forming the core of evolutionary biology by explaining how populations change over time through the forces like mutation, natural selection, genetic drift, or gene flow or migration and non-random mating where you can use the mathematical models to track changes in allele frequencies, showing that evolution isn't just about individuals but shifts in the genetic makeup of entire groups, leading to adaptation, speciation, and biodiversity. Now it comes to the genomic markers application for the functional versus neutral markers. So this also you need to understand in terms of definition just. So the functional markers directly affect the gene expression or protein expression and function. Neutral biomarkers are not functional but are statistically relevant to the diseases. So that means it could not be the primary target, but it could be the secondary source of alteration or leading to certain diseases. So this you can think in different perspectives. For example, healthy biomarkers, single biomarker, joint biomarker analysis. You can think in multiple perspectives. and you can apply multidisciplinary approaches, for example, systems pharmacological approach, network biology approach, many more things you can think about. Now, finally, it comes to the advantages and disadvantages. Of course, there are a number of benefits and limitations also, but currently there are more advantages and benefits with respect to the limitations and challenges. And most of the limitations currently is from the experimental side, not from the computational side. All types of biomarkers could be seen or analyzed in terms of advantages and limitations, which I have presented here. And you can also view it by reviewing the videos again and again for this slide specifically. So advantages include high specificity and sensitivity, stable across life, applicable across diseases, useful for early detection and prevention. Then limitations and challenges are marker trait association does not always imply the causation. Population specific variations, ethical and privacy concerns are which are minor limitations. Requires advanced bioinformatics, which is like depending on the employees involved in the project, how much they are efficient with computational approaches. So this is also not the limitation anymore. Now, real world examples of the genomic biomarkers. You might have heard again and again tumor markers, but depending on the types of cancer, different tumor markers would vary. So here there is some kind of summarized view. For example, acquired hallmarks. Then what it includes, sustained proliferative signaling, RAS, KRAS, PTEN, PI3K, TMTOR, RAF, MEK, ACK. And then it goes to the evading growth or suppression nature, RV, TP53, etc. So what is acquired hallmarks, which you could think and how you can think for different types of cancer, this is up to you. And this is the best way to present. For example, BRCA1 and 2 is very well known and well established biomarker for breast and ovarian cancer risk. Then HER2 for breast cancer therapy, APO E4 Alzheimer's disease, HILA B57.01 for drug hypersensitivity. So the tumor biomarkers for diagnosis and prognosis and targeted therapy, you need to understand all the lists of known biomarkers for specific types of cancer which you are studying or disease, you need to understand. Then it comes to the last part, that is in conclusion, what we conclude from the genomic biomarker tutorial today is basically genomic markers are the foundation tools in modern genetics, genomics, and medicine. They enable the researchers and clinicians to decode the genetic variations, predict the disease risk, guide drug development, and deliver the personalized healthcare. As sequencing technologies and bioinformatics approaches have advanced a lot and continue to advance, genomic markers will play much more crucial role for more, even for more critical roles in the future